Hi, Xiajing.
Hi, good morning.
How are you doing?
Yeah, good.
Good to see you. Do you want to test your slides?
Yeah, sure.
Thanks.
Let me see.
That's a cool coffee machine you have behind you.
Oh yeah, I need coffee every day.
Yeah.
Can you see the slides?
Yeah, looks good.
Okay. So if
Yeah, I've
if
just
this will, so if this will you see the full screen or only the
full screen. Yeah.
yeah okay.
Well, right now you stopped the slideshow, but before I saw the whole thing.
Okay. Try to see.
Yep, looks good.
Yeah, but looks like here. Would I show you that they have both the presenter show?
No, I see just the slideshow. I don't
Oh,
see your notes or anything.
okay.
Yeah, I've just been having like coffee, like powdered coffee in the morning. I don't have a coffee machine here.
Yeah, so I at least need 3 cups of coffee every day.
Same. Yeah.
And actually, this is a little complicated because you need to wash the machine every
Yeah.
time. So the Nespresso is quite an easy way.
Yeah. I bought one for my lab. It's really like the pods are very expensive, but they like it.
Yeah.
So your university still you can't go in or out?
Yes, currently is, and it looks like for the following 2 weeks.
More 2 more weeks.
Yeah, 2 more weeks, I think. This week
Wow.
and the next week. Yeah,
This
but
week.
actually,
Wow.
actually for us, it's okay because in the campus it's safer, and then we have food, we have everything much better than some people had to be locked in their building in the apartment.
Hmm. Yeah. I'm not locked in here in my apartment.
Oh, so it's called that Xiajing because more and more building will lock following these days.
Yeah, just I feel like if they're doing a lot of testing, they'll find tons of cases everywhere.
Yeah, I think especially these 2 days, these so many confirmed the case.
Hmm.
I think I worry that the school will not open within this
Yeah,
month.
I don't think the schools will open.
Yeah.
But it seems like many cases are just asymptomatic.
Yes.
Most.
But I think they worry that the all of the hospital that will be so many people.
Yeah. You could stop sharing your screen now if you want. I'll
Oh,
just have like an introductory slide first.
okay.
And your daughter's school is closed,
Yeah,
or online rather.
yeah. Right now they all just have classes online.
Yeah, same with my kids.
Yeah.
I'm getting tired of the rain already.
I think 1 or 2 more days they will be better. And I also I didn't like the rain and it's so cold these days.
Yeah, I miss California.
Oh yeah, I know.
Hi,
Hey,
左。
Xiajing.
Hi.
Hi.
Oh. Ruby was
So
here for a
you
second.
How's your how's your
How's
your
my what?
current still closed or you
yeah,
work out?
yes, we
Uh-huh.
were closed in the campus.
Okay, yeah, no,
Yeah.
ShanghaiTech, yes.
Mine's
So
not. I'm not closed.
Okay, nice. Ruby. Sorry,
Hi.
my cat.
hi.
Let me see. Okay. You can you hear me now?
Yeah.
Yeah.
Okay. Should I test the slide?
Yes, please.
Okay, thank you. All right, and you can see this.
Yeah. Try the
You see the
slideshow. Yeah. Try
slideshow.
the slide. Yeah.
Should be okay, right?
Yeah. Looks good.
Looks all right.
Looks
Okay,
good.
Yeah,
Oh,
right.
wait. Yeah. Okay. Yeah. Good.
good. Good morning.
Morning.
Early morning.
Yeah.
I heard like Congress in America voted to keep daylight saving time standard or something like that. So maybe it'll always. I don't know. I don't know
Yeah,
if that'll.
Without changing times, just keep
Yeah. Yeah.
one type of time. Yeah.
Yeah.
So I never talked to Zilong before.
yeah,
So Zilong, are you which institute are you in?
yeah. I'm in Institute of Neuroscience.
Neuroscience.
Yeah,
In the Chinese Academy
yeah,
of
Academy of
Science.
Sciences.
I
Yeah,
see. In Shanghai.
yeah, in Shanghai. Yeah.
Okay.
I visited academia Sinica like 2016. So we're
Uh-huh.
a long while
2006.
ago. 16.
2016.
Yeah,
Okay. Yeah.
yeah, it's a while ago. So yeah.
Mm-hmm. That wasn't too long ago.
6
Just
years ago.
5 or 6 years. Yeah.
Yeah.
Uh-huh.
That was a Wednesday. I suppose the museum was open for the Yinxu Museum was open. I didn't realize that before, but I realized that was the only day that open for the whole week for the museum in the campus.
Oh, okay.
Yeah. So that's
Uh,
running to the museum with a lot of tickets.
I even haven't been to that museum.
Ah.
So far, I haven't been there. Yeah. So Aaron, you come, we go together
That would
to
be
that
great.
museum. Yeah.
It was only Wednesday something. There was the only day the museum opened. That was one of the oldest Chinese dynasty called the
I think they
Indian
have like
dynasty.
oracles on the turtles
Yeah.
or something like that.
Yeah, there's a lot
Yeah.
of stuff, some bottles, something. Yeah, amazing.
And
Right.
then Ruby, when I was visiting, remember we went on a tour of the campus.
Right. That must be on the humanity side. Yeah.
Yeah, I
Where
could show
your
you.
friends, yeah.
Yeah, I'll share that screen. Let me see if I can share it. Where is it? John Nye, his grandfather, I think he was a big shot at the. So Ruby and I went on a tour, and I found this is a statue of John Nye's grandfather.
Yeah.
Really?
Is on the
That was
campus.
famous person.
Oh.
It's a very,
Yeah,
very
he's a
famous person.
very
Yeah,
famous.
Wang
Yeah,
Shiji. Yeah.
yeah, famous person. And then that was when I was visiting.
I took
And
a
then we
good
all,
picture, right?
yeah, that was a great picture. And then also the Palace Museum is really nice.
Right.
Yeah,
In Taipei, right?
yeah. And Sun Moon Lake, we went there. Yeah.
Yeah.
So the TSFN is like in early September. It's right
Right.
before China SFN.
Okay. Yeah, hopefully at the time there won't be quarantine, or maybe just 3 days, hopefully.
Yeah. 3 days
See,
would be
now
okay.
the quarantine is 10 days. It's much shorter than in China, I think.
China, it's like 3
China
weeks.
still 21 days. Yeah, that's very long.
Yeah, hopefully everything finish up this year. It's a nightmare.
Yeah. Things will be much better like next year for sure. Yeah.
That's what we said last year. That's what we said.
It's supposed to end, right? It's never gonna be good. So we start. Yeah. Always some hope.
Yeah.
Right.
All right, Aaron, could you get us started?
Yeah. Okay. Hi, everyone. Thanks for joining again. As the cases pick up in Shanghai, well, Zalong and I will still be here for you every week to get your science and interactions, even if we have to stay home. So before we get started with talks this week, announcement that next week we have more great talks. Sindori Chetty from Stanford and Chen Li from Tong University will be coming, so tune in there. Make sure to keep contacting Zalong or me and present your latest research. Okay, Xiajing is up, and Zalong, you can introduce her, please.
Okay, it's my great pleasure to introduce my colleague Xiajing Tong from ShanghaiTech. The full name is Shanghai Technology. Actually, I always said ShanghaiTech. I forgot the complete name. ShanghaiTech University. Right? So Xiajing is graduate bachelor from USTC. 中科大 is got her PhD from Shanghai Institute of Biochemistry and Cell Biology Institute, where I got my PhD. Worked with Professor Zhou Jinqiu. Now, after PhD, he went to Harvard. She went to Harvard, worked with George Kaplan, with start work use the worm, the elegans, so to start a neural system. So in 2017, she come back to China, started her own lab in ShanghaiTech. In quite a few, just a few years, Xiajing has started to make an amazing new discovery about the sexual dimorphism in the sexual synaptic transmission. Use C. elegans as a model system. Okay, today Xiajing brought her latest. So yeah, the sexual dimorphism is in the synaptic transmission. Okay, welcome, Xiajing. Can you share slides?
Okay, sure. Can you see
All
the
right.
slide? Okay.
Yeah,
Okay.
yeah, take away. Mhm.
Okay. Thanks, Long, for the nice introduction. And I also would like to take this opportunity to thank Aaron and Long for organizing such great Neuro Zoom talks. And it's also my great honor can share my work with all of you. So today I'm going to talk about the sexually dimorphic synaptic transmission at neuromuscular junctions in elegans. So as you know that the sexual dimorphism in the physiology and behaviors are ubiquitous among the animal kingdom. So in the birds that usually the male are more colorful, and in some animals that males always show a more prominent behavior in courtship, and the females usually are more involved in some of the parenting behaviors. So we would like to know what's the molecular mechanism and the circuits that underlying this phenotype. So not only for that, many psychiatric disorders also show a sexually dimorphic prevalence. That for male, they are 5 to 6-fold higher rate for the male to be affected by just like autism. And the females are prone to have just like the MDD. So if we understand the sexual dimorphism in our nervous system, it would help us to understand not only the sex-specific behaviors, and also would shed light on the pathophysiology of some of the psychiatric disorders. So we know that the animal behaviors are mediated by the reliable synaptic transmission in our nervous systems. And its disturbance also would lead to that many of the psychiatric disorders are those I mentioned, autism and depression. So here, since I started my own lab, I want to use just the C. elegans as a model to study that how the formation of the sexually dimorphic synapse that can contribute the behaviors and may better understand some of the psychiatric disorders. So here I use the C. elegans as our model organisms. So in the C. elegans, they also have the two sexes, the hermaphrodite, that is a somatic female, and also the males. They both have the shared neurons, also have the sex-specific neurons. The hermaphrodite have 8 sex-specific neurons that mediate the egg-laying behaviors. And the males have some of the male-specific neurons, usually mediate some male-specific behaviors, including the copulation behaviors. So compared to the mammalian system, it's quite simpler, just in the C. elegans. And also it possesses a very strong genetic tools that in their nervous system is very clear. So which can bring us an excellent model to study the sexual dimorphism in the nervous system. So the sexually dimorphic synapses that can be just generated by the environment. So sometimes environment can modulate synaptic transmission in sex-specific behaviors. And it also requires its genetic basis so that they can present specific phenotype only in one sexes. So the first part of my talk is how environment can modulate synaptic transmission just in a sex-specific manner. Here we focus that the function of the pheromones. So as we all know that the pheromones can function as chemical signals to modulate animal behaviors. It is very conserved in many animals. So one of my graduate students came in that he is interested to know if we culture the hermaphrodite C. elegans, the worm, in the conditions that enriched with the male pheromone, whether they would present different behaviors than those control groups that conditioned with the hermaphrodite-only pheromones. So they find actually the male pheromone environment can cause a decreased locomotion velocity in the hermaphrodites if they grow into adults. So it looks like that the male pheromones can modulate the hermaphrodites' locomotion behaviors. So next we want to study how it happens. So the locomotion behaviors are mediated by the neuromuscular junctions. And in the C. elegans, the neuromuscular junctions comprises the excitatory cholinergic synapses and the inhibitory, the GABAergic synapses. So their counteract activation mediate the sinusoidal movement of the C. elegans. So prompted by this data, we tested whether the pheromone can modulate the synaptic transmission at the MJs. So we recorded the MEPSC of the MJ in the hermaphrodites that cultured from the male pheromone environment and the hermaphrodite-only pheromone environment. And we find actually the male pheromone environment can promote the acetylcholine transmission at MJs. And we did not observe such increase of transmission at the GABAergic synapses. So from here, we get this conclusion. So we want to know how the hermaphrodite can sense such male pheromones. So the pheromones usually can be sensed by the chemosensory neurons. So in C. elegans, there are many chemosensory neurons in the head. We use the genetic ablation to ablate the individual chemosensory neurons.
What's up, bitch? Suck my dick, motherfucker.
So here we find
Sorry.
that
Suck my dick, motherfucker.
Yo, big
Suck
feet.
my dick, bitch. Fuck me, Chinese puta, fuck me. Puta, hija, puta, fuck me. Oh yeah, fuck me hard, daddy. Yeah, fuck me in the ass. Ni hao, ni hao, ni hao, ni hao, ni hao.
Indians,
Ni hao,
Asians.
fuck me my ass. Ni hao, ni
Excuse me, I run this meeting.
Ni hao,
Excuse
fuck
me,
me,
do
ni
not
hao.
unmute me.
Stop
Do
muting
not
me, you fucking
unmute me.
bitch.
That's disrespectful. As a woman, that was
Okay, sorry about that. I muted everyone.
Yeah, how about the screen? I don't know the screen goes.
Oh. Sorry about that. Let me try to stop that.
Sure.
I don't know how to stop the annotation. We've gone 2 years with this not happening, so
Yeah. You can set up from the screen.
okay. All right, let me all right.
No. Yeah.
All right, I'm gonna okay.
So maybe sometimes you can log on with some different account, but we can set the method to stop the.
Let me try this.
Yeah.
Okay. I stopped everything. Let me unmute. Xiajing, can you unmute?
yes, I can.
Okay. Can you share your screen now?
Sure.
Sorry about that, Xiajing.
Yeah, it's okay. So can you see the slides?
Yep.
Yes,
You want to
yeah.
swap display so we can see
Okay.
the
So
screen.
here, so we just
Wait. Still there.
All right. Let me. All right. Who's doing that? 1 second. Can you stop sharing your screen, Xiajing?
Sure.
Yeah, I don't know how to set up the screening writing stuff.
Yeah, I don't know how to stop let me try how to stop the annotation. The screen share menu. Okay. Let me. Okay. Let's try it. Let's try it again.
You want me to share the slide?
Yeah. Let's. Let me. I want to try to remove the annotation, but I'm not sure how to. Let me see if I can do that.
Yeah, understand. Don't know how to do.
All right, more. Disable annotation for others. Okay, I did that. Let's see if that works.
Great.
You might need to do that too, Xiajing. Maybe when you share, try sharing your screen.
Okay.
And then, yeah, try sharing your screen.
Then
go to view,
so
maybe view option. Or is there some? Yeah, just is there something where it says more and then disable?
More disable.
Is there disable annotation for others?
Right click.
Yes, I already did.
Stop share again. Yeah. Right click.
Yeah, I did.
You can stop and try it again. Okay. Okay. Yeah. Share it again.
So can you see?
Try to
try it again.
Hmm?
All right.
But okay, let's just try. We might have to disable sharing.
Sure.
Okay.
All right, let's try again.
Okay, so I continue?
Yes, please.
Okay, so here I just genetic ablated individual chemosensory neurons. Oh, sorry. So here I just genetic ablated individual chemosensory neurons by the genetic ablation, and actually I find that when I ablated the AWB chemosensory neurons, then it can block the pheromones effect on neuronal junction transmissions. So I also expressed the GCAMP in the AWB chemosensory neurons, and we find actually the male pheromones can just cause a very robust increase of the calcium present in the neurons. So which suggests that the AWB chemosensory neurons are involved in sensing the male pheromones. So So because they always try to remote control my screen.
Oh, I think you have to, if you just click on your, yeah, there you go.
Okay, so here next we want to know, because the male pheromone environment, they can modulate that the cholinergic synaptic transmission in hermaphrodites. So we want to know what's the synaptic target of the male pheromones. So in the synaptic transmission, the neurotransmission of the acetylcholine can bind to the postsynaptic receptors, that which they can just transmit the electrical signals from presynapse to the postsynapse. So here we want to know that whether the synaptic vesicles, the receptors, or the calcium channel abundance that would be modulated by the male pheromones. So we studied the synaptic structures, the postsynaptic receptors, and the presynaptic calcium channel abundance in the hermaphrodites that grow in the male pheromone environment. So here, these studies focuses our attention to the presynaptic calcium channels. So we use the split JP strategy to label the endogenous calcium channels at the cholinergic synapses. We find actually that the male pheromones can promote the accumulation of the calcium channels, specifically at the cholinergic synapses. So in order to confirm that the calcium channel is the synaptic target for the male pheromones, we exposed the UNC2 mutant that would be lack of the CAV2 calcium channel in the male pheromone conditioned mediums. Then we find that the male pheromone cannot increase the MEPSC rate in the UNC2 mutant hermaphrodites, confirmed that actually the calcium channel is the synaptic target of the male pheromones. So last, we want to study what's the physiological significance of the modulation effect by male pheromones. We find the male pheromone environment can modulate the hermaphrodite locomotion behaviors, which can promote a higher mating efficiency in hermaphrodites. So for the first part, we actually find that the male pheromones can modulate the cholinergic synaptic transmission at the neuromuscular junctions. We find this modulation can alter the cholinergic transmission and can decrease the locomotion velocity in the hermaphrodites, and also can promote the higher mating efficiency. And all of this modulation is in a sex-specific manner. So here, the first part we talk about is how environment can modulate the synaptic transmission. So, and we know that synaptic transmission also has a very strong genetic basis. So here, actually, we find that, as we mentioned, there are two sexes in C. elegans, that the hermaphrodite and the males. So actually, the two sexes also displayed distinct locomotion behaviors. The males have a more prominent curvature during the sinusoidal movement, and also the locomotion speed is much higher in males than in hermaphrodites. So we are, as I mentioned, that the locomotion behaviors is mediated by the neuromuscular junctions in the C. elegans. So here we want to know, just this data prompt us to test whether the synaptic transmission also differs in the two sexes. So we want to study whether the NMG synaptic transmission differs between hermaphrodites and males. So we use the electrophysiology to record the MEPSC at the MJs. We find that the males present dramatic higher MEPSC frequency than the hermaphrodites. And there are also a slight increase in the MEPSC amplitude, but no significant differences in MIPSC and amplitude was found between the two sexes. So the first part, we find that actually the cholinergic transmission is significantly higher in males than in hermaphrodites. So since we observe the sex difference behaviors, and we find the sex difference in cholinergic transmission. So next step, we want to study what happens in the synapse that can cause the elevated cholinergic transmission. So same here, we want to study whether the structure of the synapse, whether the postsynaptic receptors, that their abundance are different between the two sexes. We use the scarlet to label the endogenous acetylcholine receptors and monitored their synaptic abundance in the two sexes. We find actually the males have increased abundance of the acetylcholine receptors. In the elegans, there are two types of acetylcholine receptors. So here we labeled is the nicotinic acetylcholine receptors. There are another type called the levamisole sensitive acetylcholine receptors. And similar as the nicotinic acetylcholine receptors, the males also show the increased abundance of the levamisole sensitive acetylcholine receptors. But when we deletion the acetylcholine receptors, it cannot block the sexually dimorphic locomotion behaviors, because although in the mutation that lack of the acetylcholine receptors, the locomotion speed is dramatically reduced, we still can observe the sex differences in locomotion speed in the mutants. So next, we just test whether the synaptic structure is different between the two sexes. So we use the electron microscopy to study the outer structure of the neuromuscle junctions. We fix the sample, do the serial sections, and try the 3D reconstruction. Here is the way we do that, the serial section, and after that, we're using the 3D reconstruction and by this strategy to study the outer structure of the synapse in the two sexes. Then finally, we would quantify the synaptic vesicles and the dense core vesicles in the two sexes. So here we find actually the male cholinergic motor neuron terminals are enriched for synaptic vesicles. And we see a trend of increase of the dense core vesicles, but maybe the variety, we did not see the significance between the difference between the two sexes. So next we want to study what's the molecular mechanism of these sexually dimorphic synaptic vesicle numbers. So previous study had shown that the chromatin kinase 2 is required for the biogenesis of synaptic vesicles, because in the mutant that both the SV and the DCV number are dramatically decreased. So we want to know whether is the sex differential expression and abundance of the chromatin kinase 2 that cause the sex differences in the synaptic vesicles. So here we use the split therapy system to label the endogenous chromatin kinase 2 at the cholinergic synapses. So we find actually in males, the chromatin kinase 2 is more abundant in males than in hermaphrodites by showing an increase of the punctal fluorescence. So to study whether because of this sex differential expression of the chromatin kinase 2 cause the sex difference in the SVs. We just quantified the SV numbers in the two sexes in the chromatin kinase 2 mutant, and we find the mutation of chromatin kinase 2 can block this sex difference of the SV number. So further, to investigate whether actually chromatin kinase 2 can mediate the sexually dimorphic cholinergic transmission, we recorded the MEPSA in the UNC43 that's included the common kinase 2 in C. elegans hermaphrodite and the males. And also the mutation of UNC43 can block the sexually dimorphic acetylcholine transmission because the MEPS frequency in the two sexes are comparable in the mutants. Further, we also just recorded the locomotion behaviors in the UNC43 mutant hermaphrodites and males, and it also can block the increased locomotion velocity in the males. So from here, we find actually the common kinase 2, that is UNC43 in C. elegans, is an essential component for the sexually dimorphic SV abundance and can mediate the sexually dimorphic cholinergic transmission and the locomotion behaviors. So from here, we already identified the differences between the two sexes in the behaviors, circuits, and synapses. So last we asked, what's its relevance with the sex? So in the C. elegans, the two sexes shares both the shared neuron and the sex-specific neurons. We screened all over most of the sex-specific neurons, but did not find that any neurons can mediate the locomotion, the sexually dimorphic locomotion behaviors. We also screened the sex-specific process, including the oogenesis, spermatogenesis, hormones, pheromones, and neither of them mediate the sex-specific locomotion behaviors. Last, we focus our attention to the sex-shared neurons. So in the C. elegans, that the global somatic sex are determined by this pathway that composed by many transcriptional factors. So in this process, the Cha2 determines the sex of the somatic cells. So the Cha2 is a transmembrane protein. After shedding, the intracellular part of the Cha2 can translocate into the nucleus, which can promote that the males, which can block couple transcriptional factors that can promote the hermaphrodite differentiation. So as a result, in the Cha2 mutant, it would promote to the cell undergo the male-specific differentiation. So here we mutated the Cha2 in the X hermaphrodites, and we can see the Cha2 mutation can block the sexually dimorphic locomotion behaviors, and it presents just the increased locomotion speed just as the males. So in the future direction, we just constructed the neurotype specific masculinization and want to determine which sex shared neurons actually mediate the sexually dimorphic cholinergic transmission and the locomotion behaviors. So here is a conclusion. We find that actually the cholinergic transmission is significantly higher in males than in hermaphrodites. And we find actually the male cholinergic motor neuron terminals are enriched for acetylcholine receptors and the synaptic vesicles. We identified the common kinase 2 is an essential component for those sexually dimorphic SV abundance, and it mediates the sex-specific cholinergic transmission and the locomotion behaviors. In the last, by masculinization of hermaphrodites using the Cha2 mutation, we can block the sexually dimorphic locomotion behaviors. So here is our working model. So, and in summary, my talk focus on the environmental component and the genetic basis that can mediate the sexually dimorphic synaptic transmission. So in the last slide, I want to thank our collaborators from Jitao from Queensland Brain Institute, Chen Li from Shanghai Jiao Tong University School of Medicine, and Shang Bang from Huazhong University of Science and Technology. I'd like to thank all of the foundings and thank CDC and NBRP. Thanks for your attention. I would like to take questions.
Great, thanks, Xiajing, for a beautiful talk. And now we'll open questions. Please raise your hand. Questions, please raise your hand. Yeah, quick question, Xiajing. So you mean the ONC43 is the CAMK2 version of the worm version of the CAMK2, right?
Yeah.
So why is it so specific with male specific cholinergic transmission in worms? So I'm curious about but it's also we're working on some female synaptic connection, right?
Yes. So actually, we just find that they express both in males cholinergic neuron and in females, but just as the abundance, and they are more abundant in the males. So and that's because there are more synaptic vesicles in the males.
Okay. Great. Yeah. Now, sorry for the interruption. We need to raise your hand in order to ask the questions. Is that right, Ira?
I think so.
Yeah.
I think people should raise their hand. Let me see if I can allow the chat again. Okay, let me. I'll try. Yeah, people. I think you can use the chat now. Okay, hopefully things are back under control. Oh, Kai Wen. Sorry, Kai Wen, you can ask your question.
Muted.
There you go.
You can go ahead. Yeah.
Kai Wen, you're.
Okay. Yeah.
You're on.
Okay.
You're on.
Hi. Yeah. Hi, Xiajing. Great talk. So I'm curious about the sexual dimorphism. So is there because you're using C. elegans, so I'm not sure whether you can look at the developmental stage. So I'm just curious whether this sexual dimorphisms will only occurs after certain neurodevelopmental stages. So, or they are just like there's a nature part that I think there's a genetic parts that don't rely on the environmental cue, right?
Yeah, it's a really good question. So actually, just for the pheromone modulation of the sexually dimorphic behaviors, we find that the pheromone must take functions in the certain development stage. So in C. elegans, there are 4 larva, L1, L2, L3, L4, 4 larva stage. So that the pheromone functions take place in the L4 stage, and when the worms reach the adult stage, they would present the behaviors. So for the two sexes, male and hermaphrodite, in the very early development stage, they are very, very similar. So when they go into the L3 and the L4 stage, they begin to show the sex differential pattern and the behaviors.
Okay. Is any more questions? Yeah, Katie. Just a second. Just a second.
On mute.
Hi,
Xiajing.
Go ahead.
Nice to see you. I can't turn on my camera, but that was a lovely talk. I was wondering about the male locomotion behavior, because obviously male mating behavior is not just speed, it's reversals and, turning and all that kind of stuff.
Yeah.
Did you see an increase in all the different features of mating behavior, or were you just measuring pure speed alone?
Yeah, it's great questions. Yeah, actually here I only focus as a speed, because actually I studied the different parts. Locomotion, its turn and its curvature. It can be mediated by different parameters, just different genetic components. So here I only focus the locomotion, just the velocity. Yeah, because in some of the cases, some of the parameters, the turn and I find it can be mediated by different factors.
Okay, thank you.
Thank you.
Hey, so
yeah.
So CFD has a question. They can unmute.
Yeah, I'm trying to.
Yeah, there you go.
Oh, questions. Okay, I saw that.
So CFD, you can ask your question. Go ahead.
Okay.
I have a question relating to the first part of your talk, and I would like to know which specific pheromone from the males will be influence the locomotion behaviors of the hermaphrodites, and do you exact logical concentration of that specific pheromone?
Oh, so I didn't catch that. You mean what specific pheromones that the male pheromones that can modulate the hermaphrodite?
The pheromones from C. elegans, the C. elegans can produce tens of hundreds of pheromones.
Oh, yeah,
right?
yeah. I got it. Yeah. It's great questions. So actually, there are actually some of the male specific pheromones like the ascaricide number 10 that already be reported, and we tried that all of the published the male specific pheromones cannot modulate the hermaphrodite. So here we're using the LCMS try to identify the specific pheromones, but actually we only can narrow down it's just a mid because they are short chain, the mid chain and the long chain ascaricide, and it can be mediated by the middle chain hydroxylated ascaricides, but we didn't define which specific ascaricide do this job.
Great, thanks. Okay, no more questions. Thanks, Xiajing, for great talks. Like up of the second talk for Ruby.
Yeah, thanks so much, Xiajing. Amazing
Thank you.
results. And apologies for the interruptions. Zolong and I'll make sure we have things in place for next week so that doesn't happen again. And but thanks for sticking with us. Okay, so it's my great pleasure to introduce the next speaker, Yunru Ruby Chen, my great friend and colleague, and she received her undergraduate degree in National Taiwan University, then came to North Carolina where she did a PhD at NC State, and here she used hardcore biochemical studies of the kinetics of caspase folding reactions. And she developed this expertise and then brought it to UC Irvine for her postdoctoral fellowship with Charlie Glabe, where she performed kinetic measurements of protein folding to solve problems of how amyloid beta peptides aggregate and progressively form oligomeric species and then amyloids. And she made key insights into that mechanism, which obviously is important for neurodegenerative disease. She then started her own lab at Academia Sinica in Taipei in 2007. I think that's the same year I started my lab. And here she brings her chemistry expertise and approaches to tackling questions in neurodegenerative disease. She's made major contributions to studying pretty much all of the key proteins in neurodegenerative disease, alpha-synuclein, tau, TDP43, and A beta. She's made novel conformation-specific antibodies for some of these proteins and has used these to not only elucidate novel mechanisms, but also to develop therapeutic strategies. So looking forward to hearing the latest, Ruby.
Okay, thank you very much, Aaron. Should I do anything to stop the annotation? Like, do I need to do
maybe
anything?
Yeah. So
how to
go
do that?
under the view. I think under your view options, there might be something where it says annotate and there's more or something you could disable. Do you see that? Under it's in the Zoom. It's the Zoom, not the PowerPoint, but it's like the Zoom options. Actually, let me share the screen, my screen, and then I just forget how to do it. Let me just share mine, and then I'll know how to do it. So Okay. So once you're sharing, there's a thing on the right that says more, and then you can disable annotation for others. Okay. So try that. So share your screen.
After sharing.
Yeah, share your screen, and now there's like there should be like many choices up
There's
there, and one of them something that says more.
Okay, yeah. Okay, yeah.
Now go to that, and it says like disable annotation from others.
Okay, I see. All right. Okay.
Yeah, and then next week
work.
I'll make it so other people can't. I'll make it so other people can't do it, but for now that'll do it. And then just go to your yeah. So, oh, now
right,
you just want
to.
Oh yeah, stop annotating. So you want to close that annotation thing. There's like a X where you now do slideshow.
Okay. Yeah. All right. Great. Right.
Okay.
All right. Sorry for the delay, and thank very much for Aaron's great introduction. And also, I really happy to join this Neuro Zoom talk, and thank Aaron and Zilong to set this up. So I have been very amazed with the science here, and as Aaron introduced, I'm actually a biochemist, so please bear with me with the many biochemistry data today. So today I will talk to you about some protein misfolding problems. So as we know that neurodegenerative disease is kind of a dysfunction in the whole neural system from molecules to synapse transmission to neuron and circuit and a network in the brain. So there's several major risk factors in neurodegenerative disease. In general, aging is the highest risk. And then some of the disease have a genetics influence, but most of the time it's like a very small percentage, 5 to 10%. And also the environmental factors affect the risk of having neurodegenerative diseases. And I think most of you know that protein aggregation occurs in nearly all neurodegenerative diseases, if not all. And this kind of protein aggregates forming inclusion bodies in different parts of the brain. They are either in the cell or outside the cell or even in the nucleus. So many of these inclusion bodies have been characterized as amyloid. So we know that the major neurodegenerative diseases, of course, the most prevalent one is the Alzheimer's disease, which already over the whole population of Taiwan is 23 million. Yeah, and including some other dementia like frontotemporal dementia, together the world dementia case is like 47 million. And each of these neurodegenerative diseases have a very related pathogenic proteins, for example, A beta and tau in Alzheimer's disease, and TDP43 in both FTD and ALS. And these proteins were actually aggregates, and a lot of them have been characterized into amyloids. So what are actually the amyloid proteins? And this kind of proteins actually undergo a kind of protein aggregation from a natively unfolded or a partially unfolded structure, and gradually going through some oligomerization and then finally precipitate as the long stretch fibers. So this kind of fibers is usually beta strands, the secondary structures beta strand, and stacking on top of each other. Okay. Okay, but many evidence have shown that the prefibrillar species are actually more toxic, or the actual toxic species. But however, how they form this kind of toxic species is still largely unknown. So in another diagram that we show this polymerization, we can actually use a very simple dye, for instance, a dye, thioflavin T, that chelate onto the beta strand structures. So once it binds to the structure, it fluoresces. However, before this structure formation, it's silenced. So there will be a nucleation phase if we follow this SIOT kinetics, where the first oligomerization is actually without any signal, but then once the fiber grows, the SIOT signal will increase and then finally go to a steady state. So this kind of aggregation is a nucleation polymerization process. Okay, so today we'll focus on the TDP43, which the best protein that Aaron loved. And this protein has been discovered in 2006 from Virginia Lee's group. And it was found as an inclusion in the FTLD and also ALS patients. So it has a hyper phosphorylation and also some high molecular weight smearing. So in the ALS case, actually 95% of ALS patient has this TDP aggregates, and the symptoms actually somewhat overlap with the FTLD. For the ALS case, is a motor neuron disease, and it affects not only the motor cortex, the motor neuron in the upper region, and also the lower motor neuron in the spinal cord. TDP43, unlike A beta, is a cleaved peptide. The TDP43 is very essential for the physiology. It usually resides in the nucleus, governs a lot of the RNA binding activities and splicing. But in a disease state, it mislocalized into the cytoplasm, forming inclusion bodies with hyperphosphorylation and polyubiquitination. But as we thought that this is another amyloid, actually there is a question that the inclusion in the neuropathology is an thioflavin negative. It's an amyloid dye negative. So also Virginia Lee and John Trzaskowski have also claimed that this TDP proteinopathy is misfolding but without amyloidosis. Okay, so with this curiosity, we started to purify these full-length TDP43 proteins. So this protein composed of 414 amino acids with structure domains in the middle, that is the RNA binding motif, RM1 and RM2, but it has a less structured N-terminus and disordered C-terminus with glycine-rich sequence. And what we found is that this full-length protein forms amyloid oligomers, and they are toxic. So together with some other data, we also generated a oligomer specific protein we call the TDPO antibody. And we were able to stand the FTLD patients with this species in their brain, but not in the control. We are also able to immunoprecipitate this species and look under the TEM, the transmission electron microscopy, and label with the TDP antibody to show that this kind of toxic amyloid oligomers really present in the patient. Okay, so also in that story, we had a kind of a side data is that we put TDP43 into our A beta aggregation system. This is the thioflavin kinetics, where you can see the aggregation curve. Once we add the TDP from 0 to 4%, it completely inhibits this fibrillization. And when we look under the TEM, we saw all the A beta has transformed to kind of a more oligomeric structure. So we thought that there might be some influence between TDP43 and A beta. So also during that time, neuropathology study have shown from Mayo Clinics have shown that 342 AD brain donors have the 57% of them has this misfolded TDP in their brain lesions. And actually these patients has more severe memory loss and also hippocampal atrophy. So saying that this is a new kid on the block. Okay, besides that, there's also another type of patients. It's the oldest old. It's called late patients. They also have these TDP proteinopathies, including A betas and tauopathy. So together with that, we continue to focus on how TDP influence A beta and what's the role in Alzheimer's disease. So again, we play the experiment with A beta kinetics and added only 1% of TDP43 protein into this solution. And we again see that the black is the A beta alone, and the red is with 1% TDP full length. It inhibits this self-leveling signal. So as the secondary structure and some conformational antibody data. And also there's less fibro form if there is a presence of TDP43. So we ask at what stage TDP can influence A beta fibrillation. So we add this 1% TDP into the A beta monomeric state or the A beta oligomeric state where we preform the oligomers, or we add to the preformed A beta fibers. Okay, and what we see is that TDP can only influence A beta at the beginning or at the middle stage, but not after A beta forming fibrils. Also, there is a so-called seeding reaction where you can put A beta fiber seed to seed the A beta to grow faster. And during this seeding situation, TDP also does not affect the seeding reaction. So next we wonder like which domain is the one that could influence this A beta fibrillation. So we have several different variants with truncation forms. And what we found is that only the N-terminal containing ones are the good inhibitors, but only the RM domains, which are structures that is poor inhibitors, as shown in this SIOT kinetics here. The blue one is the poor inhibitor comparing to the one without TDP. So this data also confirmed by the TEM data showing that the good inhibitors have less fibro form, but the bad inhibitors has a similar fibro amount like the A beta alone. So similarly, we use the end product to load on the gel, and the aggregate you can see on top of the gel that the first one is the with the full length CDP. It has very less aggregates, and also it has an increase of this low molecular weight A beta species. Okay. So next we want to look at the interaction. So this part is very biochemistry. So I just briefly say that we use ELISA and another technical biolayer inflammatory analysis to calculate the affinity between these TDP variants and A beta. And what we found is that the poor inhibitor RM actually could interact with A beta with a relatively okay kinetic affinity. Okay, so next we come back to look at the toxicity issue. So we use this aggregates product from this aggregation assay. We treat the mice brain slides and measure the long-term potentiation. And we now have used A beta 40 or 42, which are the different isoforms of A beta. And we found the red one is the one of A beta aggregate with 1% TDP43 has the most detrimental effect in affecting the LTP comparing to the A beta alone, the blue one, or the green one, TDP43 alone. So next, we inject these species into the wild type mice hippocampus and use the Morris water maze to look at their spatial memory. And we are able to see that again, the red one is the A beta with TDP43, both 40 and 42, has the worst behavior in spatial memory. And also it spent less time in the target quadrant. So then we wanted to ask, what if we directly inject TDP into the AD transgenic mice? We use APPPS1 delta E9 mice at 5 months old, and we inject either buffer, control, or the TDP43 into hippocampus. And after 1 month, we subject that to Morris water maze. And again, we see that the TDP43 injected AD mice is the most worst behavior one. It spent the last time in the target quadrant. Okay, so also we would like to prove the interaction in vivo. So we use the immunoprecipitation for extracellular enriched fraction and also the tritone soluble fraction of this mice brain. And we here IP with TDP43 and Western blot with A beta. And we are able to immunoprecipitate A beta from both extracellular enriched fraction or tritone soluble fraction. Another thing is that we stand the IBA1 for microgliosis. And we found that TDP43 injected wild type mice or AD mice both increased the microgliosis. Okay. We also check whether it changes the oligomers assembly. And there is some changes, and especially the hexadodecamer, A beta 40 and A beta hexadodecamer has a slight increase in the TDP43 injected ones. So finally, we wanted to know like what's going on in the patient. So we have used 4 AD patients, and here is the representative images where we have a Braak stage 4 AD patients, entorhinal cortex, and we stand the A beta with the A beta antibody, and also our TDPO antibody. And we found it co-localized actually largely in the intraneuronal space, like overlap with the MAP2 staining. So actually these co-localization mostly occurred in the intraneuronal space and some in the extracellular space. Okay. And finally, we try to use the Western blot to show that we have several AD patients' brain tissues, and we first tested their TDP pathology. So we show that there are 3 patients with phospho-TDP, that's with the TDP pathology. And by staining with the A beta antibody, and after quantification, we see the AD patient carrying TDP pathology has some A beta oligomer increased. Okay, so together I would like to conclude this story is, first of all, we found full-length TDP would delay A beta aggregation at initial and oligomer state, but not at the final or the seeding reaction. And the n-terminal domain containing TDP43 actually delay A beta fibrillization, but the RM also actually could interact with TDP. And then the TDP-induced A beta species impair hippocampal LTP ex vivo and also hippocampus-related spatial memory in vivo. And TDP43 interact with A beta in vivo, alter the A beta oligomer assembly, impair spatial memory function, and induce microgliosis in the AD mice. And TDP oligomers co-localize with A beta primarily in the intracellular space in the brain of AD patients. So together with this diagram, this is the A beta aggregation. And we think that TDP, with the TDP effect, first of all, it will increase the, retain the toxic A beta oligomer time window. And also it has another effect by increasing inflammation. So with this mechanism, how we can intervene this effect from TDP-43, perhaps we can use antibody or drugs to disrupt this effect. So now I want to just talk a little bit about this antibody development. Since we have this oligomer specific antibody, we would like to first target the ALS because 95% of these patients have this inclusions. And antibody can be used as a diagnosis tool for clinical trial biomarkers or for early diagnosis. And most importantly, if we can engineer it, we can use it as a therapeutic antibody. So as people know that last year, the A-beta antibody from Biogen really is the first antibody drugs that for neurodegenerative disease, it opens a green light sign, although there's still some controversial result. Okay, so here we developed this polyclonal TDP antibody to a monoclonal antibody, and we have 11 clones. And here I'm showing that using dot plot to dot monomer and oligomer protein on the dots, most of these antibody recognize only the oligomer species. And we also do the ELISA to show the affinity. And we have higher oligomer specific antibodies, and some are like, does not differentiate, like the number 6. So then we use number 909 and number 606 to do the further experiment. So this antibody has got the US and Taiwan patent to 2035. Okay, so here I'm just wanted to show that it has some potential combining with some sensitive technology like CMOA or IMR technology to detect like really small few amount of species in either the CSF samples or in the plasma samples, where we can use the O9 antibody or the normalized O9 signal to differentiate the plasma TDP oligomers in ALS patients. And finally, I would like to just show a little bit of our in vivo efficacy test that we use the IV delivery of this monoclonal antibody O9 to a Q331K transgenic mice for ALS at the very late stage at over 1 year, 13 to 15 months. And after weekly IV delivery of this antibody, and after 4 weeks, we subject these mice to Rodaro test. And we can see a nice improvement of this green one, which is injected with the O9 antibody. And the red one is the injecting with the control isotype antibody. It has a significant improvement of this motor behavior. And also in the hind limb assay, looking at the rear limbs, it also has a nice improvement, significant improvement. And most importantly, it has improved the survival of motor neuron in the spinal cord section to a very nice amount. Yeah. Okay, so with that, I think the take-home message here is that we would like to suggest that manipulating the TDP oligomers could be a potential therapeutic way for both AD and ALS. So together with that, my lab is really a protein lab that we work on many amyloids and A-beta and TDP and dipeptide repeats. But by doing this, we can try to understand the disease mechanism and develop some translational medical research. So with that, I thank you very much for your attention, and sorry for taking a little bit longer time. And I think most importantly, more important than science is world peace. Okay, and then also including the internet attack. Okay, so I think better science can overcome, this kind of bad things in the world. And you're all very welcome to come to Taiwan. We have so many good foods to enjoy. Thank you very much.
Thanks, Ruby. Awesome talk. Oh, yeah, that's the best part. Okay, we have time for questions. I wanted to first ask the effects of your different TDP43 constructs on A beta fibrillation. Did that depend on RNA binding, or do you think RNA somehow affects this?
Yeah, that is awesome question. Actually, we have some data showing we have actually did some structure studies. So we have a TDP R1 RM domain and the A beta binding complex after modeling, and the interaction interface is opposite to the DNA binding interface. So we don't think the binding of, well, it might affect, but it's not like directly blocking the RNA binding. Yeah.
Okay, now we have Haiyang. Do you want to ask your question? Haiyang is world expert in TDP43.
Not as expert as you guys. Yeah. Very nice talk. I have actually one question, and then Aaron actually stimulated me for another question. So first question is that, can the A beta aggregates in the transgenic mice promote the spreading of the TDP fibrils? When you inject TDP fibrils into the mice, did you check whether it can spread from one neuron to another?
We haven't checked, but because we only inject very small amount of TDP, like 2 microliters to the mice. So I think this transgenic mice, AD mice, it has much more amyloid than the TDP. So we didn't track the TDP after injection. But in our previous experience, we did inject like poly-GR, the dipeptide repeats, into the mice. It will be degraded very soon. So I think the injection might not initiate some spreading in this mice. Yeah,
I see. Actually, the second question is about the RM domains purified from bacteria. I heard rumors saying that when you purify RM domains, it's really hard to separate the RNA from bacteria. So you actually you co-purify it with bacterial RNA. Did you check whether it is there? Will that affect your crystal structure?
Right. Actually, when we purify, we did add our RNAs, right?
Yeah.
And after that, we check the absorption spectrum. There's no 260 left. Yeah.
That's
so
nice.
Supposedly, I think it mostly, and also after purification, I think there's less or no DNA contamination. Yeah.
I see. Wonderful. Thank you.
Xiajing. Wow, another world TDP43 expert.
No, I mean, this is fantastic work. Very beautiful work and talk, Ruby. Congratulations. I'm still very confused. The two are in different compartments, right? TDP still that intracellular or they're cytosolic, but A beta is, but presumably they still share within the endosome or whatever. They two typically don't meet each other normally. So I still have hard time trying to understand how does this interaction work. But to think about more deeply, I mean, this also open up a lot of possibility. Could they actually somehow get into autophagy or release or get to mitochondria? It just opened up a lot of possibility if you think about how they actually interact. So I want to get your thought on this.
Right. I mean, that is a very important question. But actually, at the beginning, we thought they must have, maybe TDP was split out from the cell and they interact outside of the cell. But actually, when we get this data, they are mostly colocalized in the intracellular space. We dig back to the previous literature, and we found there's many data showing that A beta is generated intraneuronally. Of course, there is a question whether A beta is in the lysosomal compartment, the endosomal compartment, or it's in the cytoplasm. I think there's both data showing, of course, A beta is generated in the early endosome by APP, However, there's also A beta going to the mitochondria, affecting the mitochondrial function or binding to the mitochondria proteins. So I think there's some free A beta in the cytoplasm, and also there's A beta in the lysosomal compartment, and TDP also could aggregate and go into the lysosomal compartment, and it is possible that they meet there. Actually, we had a kind of preliminary data shown in the AD patients. Both of these proteins are in the lysosomal compartment. Yeah, so maybe they meet there. Yeah, we don't know.
Okay, maybe I'll just read the next two questions. One from Richard Marglin. Can your method be used to investigate, your fibrillization method be used to investigate temporal dynamics between cytoplasmic TP53 oligomers and aggregates? Are they in equilibrium?
Temporal dynamics between cytoplasmic
TP53 oligomers and aggregates. Okay.
Temporal
and then maybe while you're thinking of that one, just like how long it takes, the timing between going from oligomers to aggregates.
Oh, in the cell.
Yeah.
I guess so. I mean, if we have a live cell monitoring and we have a proper dye to monitor this thing, and for example, like FCS or something, you can measure the particle diffusion time, and with that, I think it is possible to look at the cytoplasmic TDP aggregation. Are they in equilibrium?
Okay.
I think not. Usually this aggregation is like a irreversible process, and there might be some equilibrium, but at the end it must be large aggregate more than the small oligomers. Okay.
Okay, and then Stuart Lipton points out A beta sticky, is the binding to TP53 saturable and showing true specific binding kinetics?
Yeah, it is a good question. I mean, A beta is sticky. Actually, TDP also sticky. So whether this is real or not, I mean, I think it's real because when we do the IP, it does precipitate down, and also it shows the co-localization in the patient's brain. But whether the affinity, yeah, I think the affinity part is, we also have question because the affinity looks very high at 10 to the minus 7 molar. It's quite sticky. Actually 10 to the minus 12 in that biophysics measurement, and that it's part of because it's difficult to get off. The off rate is it's large. So the binding kinetics is a reference, right? I think the sticky part also contribute to some of the high affinity, but maybe the exact affinity is not that high. Yeah.
Okay. I guess that's it. Thanks, Ruby. Exciting data,
Thank you.
especially the in vivo stuff at the end looks encouraging. All right. Thanks, everyone. Thanks.
Thank you, Aaron. Thank you,
Xiajing.
Zhu.
Thanks, Ruby.
Thank you,
Yeah,
Xiajing.
sorry.
Thank you.
Yeah.
And,
Bye.
see everyone.